Hello, everybody. Thank you for attending this course today. The subject will be the virtual brain cloud, and it connects very nicely to where the previous lecture by Svenja ended. So it's about modeling, brain modeling. And as you all probably are aware of, cognition and also consciousness are relying on the dynamics in the brain, and the dynamics evolve across many different scales. As you can see here on the left, we have the molecular scale, synaptic levels, circuits, up to the system. And this is complex. This is a complex subject. The good news is that we do not actually need to seek for specific mechanisms on each of these scales or for each of the physical and chemical and biological processes. It's sufficient to look at the generic behavior of complex dynamic systems. So we are doing brain network modeling, and we try to understand the primitives, the dynamic principles that lead to the dynamics in the brain that underlie cognition, cognitive function, but also dysfunction. And for this, we combine mathematical computational modeling with imaging, as you have seen in the previous lecture. So we use the individual information of diffusion tensor imaging, for example, to reconstruct the fiber tracts and to construct brain network models. And to make sure that they are not just like static atlases or models of the brain, but really dynamic. We have to introduce dynamic models. We really seek to simulate the actual physiological entities like firing rates, membrane potentials, bolt time series, and so forth. And we want to discover the hidden physiological mechanisms that underlie these processes that we can observe with invasive or non-invasive methods. So typically in brain network modeling, one region is represented by such a population model, and this contains millions of neurons. And for simplicity and in order to get a level of abstraction, we are not simulating these networks as spiking neuron networks, as you can see here. So this is too much detail. We cannot really capture this, but we're simplifying that towards mean field or neural mass models. So each brain region is represented by such a population model that captures the main behavior of a large population of neurons, and they're interacting and influencing each other through the reconstructed anatomical skeleton, as you can see here. And a tool to do this is the virtual brain software, an open source software that has been implemented and integrated tightly in the EBRAINS platform, as I will show you later. That provides a nice framework to simplify this quite complex process, to integrate all these different pieces of information, and also to allow to modify the dynamical models to look for different aspects of brain activity. So this is the mathematics that is at the heart of the virtual brain simulator. This is an evolution equation that provides the activity, the dynamics, as a function of the activity in a brain region, in a local node, we call it, and the input from all the connected brain regions that you see here, connected through the long-range axonal connections that we reconstruct from diffusion tensor imaging data, plus, if you would like, the local connectivity, the input from the adjacent populations that you can see here, that have horizontal, lateral connections. And you can also introduce a stimulation, an input current that mimics internal stimulation of the brain or external stimulation. And here you see the noise term. So if we want to do really detailed simulations of the surface of the brain that take into account the geometry with all the gyruses and sulci, then we actually have to introduce the lateral connectivity, and we model the lateral connectivity with such kernels that provide us the information how the mutual influence falls off as a function of the distance. But it is also possible to neglect the cortical geometry and just do the modeling based on the large-scale connectome. And this is computationally much more efficient and much more widely done. So this is now the graphical user interface of the virtual brain simulator. You have a programmatic interface and a graphical user interface. This is a simulation cockpit. You see, you have many settings, but most importantly, you can select between different population models, dynamic models, so you can decide what kind of dynamics are actually captured in each brain region. And depending on your scientific question that you want to solve or the disease you want to address, there might be different choices optimal. I will show you a couple of examples later, what the different, what is behind the different names of the models. But this is a simulation, so you see that we are simulating here neuronal dynamics, we have the time series, and we have maps similar to what you have seen before. So basically the outputs of the simulator are exactly the same measures that we otherwise would get from experimental data. This includes the firing rates of neurons and local field potentials that you would typically measure invasively, up to the non-invasive measures of EEG, MEG, or fMRI. And here I show an example of my own brain. You see the parcellation, and here you see the projected simulated EEG. It's projected on the skull, and in a moment you will see the simulated fMRI activity in the brain. So here you see actually the functional connectivity and the functional connectivity dynamics that you have seen previously. And I just note when we restarted the session, I obviously took the old presentation with more slides. I wanted to skip that because we have not so much time left. But since it is showing up, I just go through it. So the functional connectivity gives you the degree of synchrony in the brain, the correlation, the pairwise correlation between brain regions. And it is stable and static if you have a long time window. So if you are recording functional dynamics over 20 minutes or more, then you will have a reproducible matrix. If you record it at shorter times, like 6 minutes or even seconds, then you will have dynamic functional connectivity. There's a switching of the degree of synchronization and desynchronization of the networks of the brain. And this can be captured in the so-called functional connectivity dynamics matrix that was introduced in the pre-final slide by Svenja, that gives you the degree of correlation between different functional connectivity matrices. So here, if we take row 20 and column 30, then, for example, this would give you the correlation between functional connectivity at time point 20 and at time point 30. And you will see that there's such a checkerboard structure. There are actually blobs that form and disappear, and this shows you how different networks in the brain are synchronizing and desynchronizing again. And these are attractors, so-called attractor states in the brain, where you get the synchronized activity that are actually underlying the flow of cognitive function in our brain. And what you see here is the simulated result. And the axes here show you the one free parameter that we can choose. This is the global scaling parameter that rescales the degree of how the different regions are interacting. And now you see that suddenly we get similar patterns as we have in our empirical data. And this is only for a small range of this free parameter. And this is how we optimize our models. We change the values of the parameters and try to identify the subspace of the parameter space that gives us the observations that we have made. So here we see that for functional connectivity and functional connectivity dynamics, they are actually converging. So it's the same space. And here we have an EEG measure that I will not explain in more detail, but we also capture actually empirical features that we measure not with fMRI, but with EEG at the same time. So now, how can we really use such simulators to understand the mechanisms? If we want to understand aging and disease, we need, first of all, to understand the healthy brain. And this is what we did in the study. We virtualized the brains of 15 healthy individuals. They had simultaneously acquired EEG and fMRI data. And here we used the so-called reduced Wong-1 model. You will find it in the simulator. We have here the input currents that drives the excitatory and the inhibitory part of each population, each brain region. This translates to the firing rates, R for excitation and inhibition, and the synaptic activities. And the firing rates underlying the resting state fMRI activity that we are simulating, that we are comparing to the measured activity of each individual. Here we see the reconstructed firing in each brain region. For the excitatory and inhibitor population, we picked here an occipital region, but in principle, we have this information available for any region. So we can infer through modeling, through reproducing what we can observe, the underlying neuronal interactions that can give rise to the observations. They can be unique, or there can be multiple scenarios that all provide the same large-scale observations. And this is also subject of research. But at the end, what does it give us? It's a comprehensive, self-consistent model of the brain that actually spans multiple time scales. So here in the upper panel, you see our simulation results. So these are the signals of a single self-consistent brain model with fixed parameters, no changes of the parameters. And the signals have been analyzed, so different types of signals, the firing rates, the local field potentials, the Bolt signal, the alpha, the EEG rhythms, activity, and compared to what we see in different empirical studies. So for the non-invasive measures, we refer to human studies in the experimental part, and for the invasive measures like firing rates or local field potentials or input currents, we refer to animal studies. And we can now look, for example, in our model simulation for the relation of the firing rates and a single 10 Hertz oscillation, the so-called alpha rhythm. So there's a very clear relationship between firing and the phase of this alpha oscillation. And this is known from monkey recordings as well. So there's the same type of relationship in experimental data. And this is encapsulated here in a large-scale model that at the same time now reproduces this relationship, but also at a very long time scale reproduces switching of functional connectivity dynamics that we observe, and many levels in between that I have no time to comment on are captured as well. And with this, we get the mechanisms, the relation between the many different observations that typically are represented in different papers, and often they come from different species, from different methods. Now they can be linked together through the mathematical equations, and it becomes clear how they are related. This is how we try to understand the brain. It lies in the nature of the virtual brain that it can be used for clinical applications because it can take into account the individual aspects of the brain. And this has been done in many different studies. So for example, there are studies on epilepsy, and there's actually a clinical trial ongoing that puts TVB to test whether it helps to make predictions what the best entry point to neurosurgery in drug-resistant epilepsy is. And this is an ongoing clinical trial where we will have the results in 2 or 3 years available. There are publications out on how the virtual brain is able to capture the altered dynamics in brain tumor patients, how it captures the dynamics after resection of the brain tumors, how the parameters, the values of the parameters in the brain change with different genetic variants that are thought to underlie brain diseases like psychosis, or how actually the parameters that we infer from our models can predict the potential for recovery in stroke or differentiate different trajectories for dementia. So there are many possibilities. I only mentioned a few, but there's always the same challenge that the degree of resolution of our models ends with a mean field approximation, the population models that, as I mentioned, typically represent brain regions that contain millions of neurons. And in some cases, this degree of resolution might not be sufficient. And to understand what the degree of resolution is that we need, we have developed multiscale co-simulation in EBRAINS over the past 2 years that allows us to place some more detailed network models in the full brain network model of the virtual brain. And with this, we can co-simulate the coarse scale at the same time with a microscopically detailed spiking network in some of the brain regions. And we can see how the detailed network interacts with the large scale activity and vice versa. And we have two use cases that are far progressed. One is done in collaboration with Alice, actually, who gave the introduction, and the Egidio D'Angelo's team in Pavia, where we make use of an existing excellent implementation of the very detailed model of the cerebellum with nest simulators, spiking neuron simulators. And this one where we made use actually of an already existing basal ganglia model that was implemented with another simulator, Anarchy, similar to nest, but it was also pre-existing. And in collaboration with a team of Anarchy around Fred Hanke, we were connecting the basal ganglia model to the virtual brain model. And I will give you here a few insights on what that brings us and why we are doing this. So what you see here is the basal ganglia model with much more detail, so implemented with spiking neurons. As you can see here, there are hundreds of neurons implemented in the model. We capture the striatum, the globus pallidus externus, internus, the thalamus, the subthalamic nucleus. These are structures that are relevant if it is about the treatment for patients with motor disease, like Parkinson's disease or also dystonia. So the goal is to improve the planning of the deep brain stimulation for those patients, because we know already from the previous work with our collaborators at the Charité, Andrea Kühn and Andreas Horn, that the location of these stimulating electrodes is very relevant. So it makes a difference whether they hit one part of a fiber, external fiber bundle or the other. And with our simulations, it is now possible to even make more precise predictions, not only of the fibers that are hit, but also the implications in terms of the dynamics on the entire brain. And therefore, we here stimulate the brain artificially on the computer. So we have a stimulus that is very similar to what is being used in the clinics, this one. And then we have a monophasic stimulus to compare to other modeling work where this was typically used. And here you see now that what you probably will never see or have never seen before for the virtual brain, that now we can not only simulate the whole brain, but some regions have these spike trains. So they provide detailed information about the spiking behavior of these nodes that are relevant for Parkinson's disease. And here I just want to draw your attention to the bottom row, and this is the thalamus. And we have here in the first column the firing rates for a healthy control, and in the third column for a patient with Parkinson's disease from our simulation. And we see that in our simulations, the firing rate for the patient and the thalamus are decreased. And this is actually also the case in real Parkinson's patients. So now in our virtual stimulation, we see the effect of different kinds of stimulation on the firing rate in the thalamus. And we see that it is getting more or less normalized to the level of the healthy patient. The nice additional effect of embedding now the spiking neural network of the basal ganglia into the full brain model is that we can also see the effects on a larger scale to the entire remaining brain. And we know that deep brain stimulation can also alter cognitive function, has differential effects on the motor function of the patients. And here we see how, for example, the stimulation with a monophasic stimulus to the subthalamic nucleus or biphasic stimulus evokes differential cortical patterns. So here we see a deactivation of the premotor region, while here with a stimulus that is typically used in the clinical setting, we have a strong activation. And the hope is to use brain modeling to virtually plan therapies, not only for motor diseases and deep brain stimulation, but in general to know the primitives of these complex dynamical systems, and therefore by knowing them, know what are the right levers in order to push the brain to the desired healthy state. And for this, the EBRAINS actually provides a full range of different tools that can be used And the virtual brain is just one tool of many. As you can see here, there are data and knowledge services, atlas services, many different simulators that are being curated in EBRAINS, brain-inspired technologies, and medical data analytics. The virtual brain cloud is a particular facility hub that allows to do the brain simulation in the infrastructure of EBRAINS, taking into account that we are dealing here with personal data. And there are many different workflows involved actually in brain simulation, personalized brain simulations. So we do not have only the main simulator, the virtual brain, that you can also get from the virtualbrain.org website, but we have the co-simulation tools, we have optimized code for high performance computing, we have the possibility to invert a model to fit the parameters and determine the parameters. We have, importantly, the processing pipelines that generate the structural connectomes, the functional connectomes, or region-specific time series that we need to construct our models or calibrate our models. And then we have also simulation-ready data that have been processed before and that are fully annotated and are made available for modeling work by the community. And this is how this looks like. Svenja had a nice workbench figure at the end of her lecture. And this is a more technical figure, but it shows the same thing. So this is EBRAINS, and these are the modeling virtual brain services and how they are connected to the core services that are already provided by EBRAINS. And this is quite enormous. So what kind of tools are offered by EBRAINS to the neuroscientists to enable collaboration and joint work on data. So this is here the digital atlases. I will go there in a little bit more detail in a moment. There is Drive, so there's a possibility to store your own data on EBRAINS, to upload them and to provide access to selected people. We have the possibility to build sandboxes or workbenches, workspaces for yourself and your collaborators. So only selected people can enter these workspaces and collaborate there in a protected environment. There are possibilities to work together on documents, programmatic interfaces via Jupyter Lab and Jupyter notebooks that you can either create online in the EBRAINS cloud, or that you can just upload if they are pre-existing, or many do exist there already that can be reused. And you have the possibility to install or use virtual machines. So for example, the virtual brain can be directly access the graphical user interface via virtual machine, and with a single click. And if you have an account for EBRAINS, you get the simulator that you otherwise would run on your computer, on your local machine, and can use the infrastructure of EBRAINS to run your simulation. You don't need your own computer for that. And then the knowledge graph service is extremely important. It provides the possibility to search for data sets, but also for models and for software that are also being curated in EBRAINS and made findable and discoverable. And then, since the computations are heavy for the simulations, but also for the processing of the imaging data, if we reconstruct the connectome, for example, we have the high performance computing backend. And this is all connected via APIs. Our services, virtual brain services, are connected, and via these APIs, there are different types of deployment that can be used depending on the taste of the user. And importantly, we have data protection mechanisms in place. So you need to authenticate yourselves as access control, encryption. The data are all the time encrypted. And when they are decrypted during the processing, they are being sandboxed. So any unwanted information flow and access by others is prevented. So now I mentioned already the atlases. They are a very important resource of EBRAINS. And there is recently has been developed the API for accessing those atlases that provide information, for example, about the receptor densities, yeah, that can be found in different brain regions. So presently, these are, I think, 35 brain regions and 14 different receptor densities. This is highly relevant also for the brain modeling. So here we just pulled through the API that is now available, the data that are available on EBRAINS in such a 3D brain model. And if you look at the hippocampus, for example, we see now the distribution of the receptors in different regions of the hippocampus. So CIA1, 2, 3 are different hippocampus regions. And we see now here on the right, the legend, there are, for example, 2 types of serotonin receptors. They are shown here in green. And one has an inhibitory effect, another one has the excitatory effect. So if we want to model altered neuromodulation, for example, this is happening in Alzheimer's disease, we need to know the ratio of the receptor that has an inhibitory and the receptor that has an excitatory effect. And it is inverting actually in different hippocampal regions. So for the modeling, this is highly relevant information because it would have contradictory effects if we would do that in a wrong way. Another important resource are the pathway inventories, the knowledge bases that use literature mining to find all the signaling cascades that are underlying brain diseases. And the Skyview is such a literature mining tool that has created databases of annotated knowledge. And based on this, pathway inventories have been created that also contain relevant information that can go in our models, because they change actually the parametrization of the models. And I will show you in the moment how this changes the dynamics and can explain the altered dynamics in patients, for example, with Alzheimer's disease. But this is just to give you an example how it's possible to put search terms in this knowledge base. And actually, there's also an ontology that provides anatomical labels. And this ontology, Oberon is the name, has been mapped also in the 3D brain. This is a cluster parcellation, one parcellation of many different parcellations that are available. We have in our tool 45 different parcellations that allow, with this knowledge base adapter, to retrieve information, for example, about the distribution of beta amyloid or tau protein or altered glucose metabolism in the 3D brain model. So this is just an illustration, again, in the 3D brain of these altered pathways and how they are mapped on the 3D brain. Now I want to show you how we integrate this information in the modeling. And I show you this with an example. I come almost to the end, so no worries, of PET data that give us the beta amyloid distribution in the brain. And we know from our pathway inventories and knowledge bases that in Alzheimer's disease, there's actually a suspected altered pathway underlying the beta amyloid in the brain, which leads to an accumulation and increase of concentration of amyloid beta plaques. And in the vicinity, this is also known from the literature, animal research and in vitro cellular research of these plaques, the function of the inhibitory interneurons is impaired. And this leads to disinhibition and the increase of excitatory interneurons. And all these cause and effect models can be implemented in our full brain network models. So here I give you another example for a population model. It's the so-called Janssen-Ritt model that captures the activity of inhibitory interneurons, of pyramidal cells, and of excitatory interneurons. And we see here now the so-called phase portrait of phase space that is spent by the activity of the pyramidal cells and the inhibitory interneurons of one population in the brain in dependence of the network input. So the population that you just have seen on the previous slide is embedded in a network, and depending on the input from the network, it can exhibit different repertoires of activities. So this is called a limit cycle, and this is called a fixed point. What it means is, see it here on the next slide, that these trajectories translate into oscillatory activity, the local field potentials that we are simulating. And if we change certain parameters, control parameters, the geometry and the repertoire of the dynamic population changes. So this is a coupling between the inhibitory interneuron and the pyramidal cell that is a function of the beta amyloid load, as we know from the literature. And when varying the beta amyloid load, the local field potentials change their frequency. As you can see here on the right, there's a slowing of this yellow trajectory. And in this way, we can actually link the altered molecular pathway of beta amyloid to the observations that we make on EEG, that is a slowing of the EEG frequency. This is captured here. So we see 3 different groups: Alzheimer, mild cognitive impairment, and healthy control. Here are the frequencies of our simulation, and we see a slowing by just integrating the beta amyloid in our models in the theta range. Usually you would not find such a peak. And now at the end, I want to show you that it's possible to actually also study virtual drug effects on our virtual brains. This is a simulation of Memantine, an NMDA antagonist that is also given in dementia. And we modeled it by modulating the impact of exhibitory interneurons on pyramidal neurons. And we could show that by providing virtual drug therapy with Memantine, we can normalize the traces of the Alzheimer patients with a slowing that I have shown you before in the simulated EEG to the normal range of healthy controls on people with mild cognitive impairment. So I think I have shown you that there are many possibilities of integrating information in brain network models, refining them with information from atlases, from knowledge that is being collected in EBRAINS and is curated and annotated and made available for researchers. I'm really curious what you will do with all these possibilities. I would like to thank you very much for your attention. I also would like to thank, of course, all the collaborators from the different consortia and my team in Berlin at the Charité.